Hello students, welcome back to the biology class. So in the last class we continued the 19th chapter, that is the excretory products and their elimination, and we discussed the topic of urine formation. We learned that urine formation is also called as uropoiesis and it has three steps. The first step is glomerular filtration, the second step is the tubular reabsorption, and the third step is the tubular secretion. We learned in the last class that the filtration process happens because of the flow of the blood with a particular value of the pressure is there, which passes through the arterial, or you can say which passes through the glomerular arterioles or through the capillaries, and this pressure is 75 mmHg. We learned that there are some negative forces also which causes the glomerular filtration pressure to decrease to down to the value of 25 millimeter Hg, and that is because of the osmotic concentration of the proteins in the protein, interstitial fluid pressure, and the renal tubule content pressure. These pressures cause the value of the pressure for the glomerular filtration to decrease. We learned in the class that the ultrafiltration occurs through the filtration membrane, which is made up of endothelial cells of the blood vessels and the basement membrane attached to them, as well as the basement membrane which is attached to the visceral layer of the Bowman capsule. We learned in the last class that there are special cells in the endothelium which carries the holes called as fenestra, that is by fenestration, and the size of these holes is 50 to 100 nanometer. And in the same manner, the visceral layer also carries special cells called as podocytes, which carry the slit pores with the size of 25 nanometer. So the real amount of filtration occurs is through the two barriers, which are the two basement membranes. We also learned that this under the net pressure of 25 millimeter Hg, filterable components passes through the filtration membrane and reaches into the lumen of the Bowman capsule. We learned in the last class that 1100 to 1200 milliliter of blood is made to pass through filtration through the two kidneys per minute, and this accounts to somewhere around within the 4 to 5 minutes the whole blood is filtered once through the kidneys, both kidneys together. So the filtrate produced per minute is 125 milliliter, and that accounts to 180 liters of the filtrate produced daily. But we all know the amount of the urine we pass is just 1 to 1.5 liter per day, so that means almost 99% of the filtrate is again reabsorbed. So we learned in the last class that filtrate carry water, sodium, potassium, calcium, magnesium chloride, phosphates and glucose, along with amino acids, hormones, vitamin, urea and uric acid. Filtrate generally never carries the fats, proteins, WBCs and RBCs. During some abnormalities or disorder situation, these compounds like proteins or otherwise WBCs, RBCs may leak out into the filtrate. We also covered the second step, that is the tubular reabsorption. We learned that because filtration is a non-selective process, so along with the wasteful substances, even the useful substances or molecules are also passed out. So therefore, 99% of the glomerular filtrate is supposed to be taken back by body by absorption, which could be active or passive. Active, which works against the concentration gradient with the expenditure of energy. Passive, which works along the concentration gradient and does not uses the energy. And because this process takes place in the renal tubule, it is called as tubular reabsorption. This reabsorption occurs in PCT, loop of Henle as well as the DCT. We learned in the last class that the main site for reabsorption is the PCT, where a large number of microvilli as well as mitochondria is there, accountable for almost 66% of the reabsorption. And we learned the various molecules and their degrees to the percentage they are supposed to be reabsorbed by the body, and this reabsorption changes the filtrate into isotonic filtrate. After this, the next level of reabsorption occurs in the loop of Henle, and it has two portions, the descending limb and the ascending limb. We learned in the last class that the thin portion of the descending limb is permeable to water. So water is reabsorbed by osmosis. This causes the filtrate to become hypertonic once again. And then the ascending limb is impermeable to water, but it allows through its thin as well as thick portions sodium chloride to be absorbed, which causes the filtrate to become hypotonic again. And the DCT is the last portion where under the influence of aldosterone and ADH, that means DCT is supposed to function only in special conditions. Otherwise, absorption does not take place in DCT. And under the influence of aldosterone and ADH hormones, sodium is absorbed, chloride comes in, and bicarbonates come in. And we learned that even in the collecting tubules and ducts, under the influence of these hormones, more permeability for the water develops, because of which hypertonic urine is secreted out, or you can say the absorption of water takes place. We learned that some amount of urea is also diffuses out of the collecting duct into the medulla, making the medulla hyperosmotic in nature. We learned that after this, tubular secretion takes place, which is an active secretion of metabolic waste from the peritubular capillaries as well as interstitial fluids into the lumen of the nephron. And we learned that it takes place in the in the PCT, but the maximum amount takes place in the DCT portion, and a small amount of it also takes place in the thin, or you can say the ascending limb of the loop of Henle, and descending limb of the loop of Henle, there is no secretion taking place at all. We also learned that there is no secretion taking place in the portion called as the collecting ducts.